Hi, everyone. My name's Brenda. I am a graduate student in Dr. Lisa DeVetter's Small Group Horticulture program. And today I'm happy to share with you my work on enhancing the breakdown of soil biodegradable plastic mulch films. So as you all may know, plastic mulch films are used widely today, and their use is expected to continue rising in future years, which is not surprising because they provide many benefits. However, there are multiple associated drawbacks to using PE mulch, which has generated multiple concerns. For starters, it's really expensive and laborious to remove PE mulch at the end of the season. It also generates a lot of plastic waste, and problems arise when this waste translocates into our environment. So to mitigate this, there's been a renewed interest in the use of soil biodegradable plastic mulches known as BDMs. They have been around since the 1990s, but improvements in research and technology have allowed for the production of more reliable BDMs today. So as you can see from these photos, BDMs look similar to PE mulch, and they can provide similar benefits. But unlike PE mulch, they are designed to be tilled into soil at the end of the growing season, where soil organisms can break it down and convert it into water, carbon dioxide, and microbial soil biomass. So this reduces the cost to growers and also the plastic waste generated. For BDMs to be suitable alternatives to PE mulch, they should not only perform similarly, but they should degrade as they're designed to. Product standards have been established to ensure the integrity of performance of these products. And one in particular that I'd like to point out to is EN 17033, which specifies that BDM should achieve at least 90% degradation within 2 years of being incorporated into soil. BDM research has demonstrated that overall BDM breakdown is generally faster in regions with higher temperatures. So this is concerning for places with cooler temperatures and in field sites that have soil which are prone to being dry or saturated. And of course, you may be thinking, well, this is concerning for many places in Western Washington. In a recent study done in Mount Vernon, Washington, degradation of multiple BDMs was assessed. And the findings concluded that realistically, it may take more than 2 years for the BDMs to achieve at least 90% degradation. And this was due to slower degree day accumulation, which is much faster in the laboratory settings. In a different study, the findings were pretty consistent, and 5 BDMs were used in raspberry production. You can see here that 6, 12, and 18 months post-burial, the degradation was pretty slow in the soil. So this brought up one very important question. How can we enhance the breakdown of these BDMs? So the objectives of my study were to evaluate how different BDMs break down under our climatic and environmental conditions. The second objective was to assess how the application of different products influence surface deterioration. And the third goal was to determine whether these products had an influence on in-soil degradation after till down. This experiment took place in Mount Vernon, Washington, and I followed a randomized complete block split plot design. The main plot represented the different BDM mulches used. The split plots referred to the products that were applied, and the split plots were used to differentiate between the timing of the product's application. The mulch treatments I used were 3 plastic BDMs, BASF 0.6, and 2 Novamont treatments which differed in thickness. I also tested 1 paper BDM which served as the control. I'd like to quickly point out that the difference from the treatments are that BASF is made from PLA and PBAT, while the Novamont mulch treatments is made from starch and PBAT. The products that I applied included Octogrow liquid humus, Wheat Farm, compost tea, and compost. The first year I used compost tea, but I had difficulty sourcing it the second year, so we switched to dairy-based compost instead. And of course, we had plots that were not treated and served as the control. The methods that I followed included establishing the mulches in the spring of 2019 and 2020. the products were applied within the assigned split plots on the surface of each BDM. And the application timing took place in the fall and then the following year in spring. For data, I collected soil temperature, soil moisture, and percent soil exposure known as PSC to measure surface deterioration. So for surface deterioration, as you can see in the photo, I looked at each split plot and assessed how much of the soil was exposed over time. And this was done in increments of 1% from 1 to 20 and increments of 5% after until 100% PSC was observed. For in-soil degradation, I took 2 samples from each plot and placed them into mesh bags. The mesh bags were retrieved 6 and 12 months post-burial, and then assessed. The Mulch samples were also taken photos before and after burial, and I assessed if there was a change in surface area over time. Now, for the findings of my study, I'd like to quickly mention that there was no effect of application timing, so I will start with soil moisture. From this table, you can see that soil moisture was pretty consistent between both years and pretty similar between the mulch treatments. The main takeaway message is that soil moisture was not a limiting factor to BDM breakdown. Now, when it comes to temperature, temperature is very important for BDM breakdown because it directly influences whether the BDMs are susceptible to microbial degradation. So in this table, you can see that, again, the temperatures were consistent between both years, and it was pretty similar between the BDMs as well. However, the temperatures were the highest between May and August, but they did not reach the temperature threshold for breakdown of PLA. So it is probable that temperature was a limiting factor to the breakdown of the BDM made from PLA. Now for PSC, the findings were the same both years, so I will just discuss findings from the first year. These mulches were established in May of 2019, and deterioration was pretty minimal between May and mid-September. By mid-September, the PSC changes started to increase for all the BDMs. And by April of 2020, WeedGuard Plus, of course, reached 100% PSC. And that was followed by Novomont 0.6, then BASF 0.6. And lastly, Novomont 0.7 showed the lowest change in PSC over time. In terms of product efficacy, none of the products that were applied had an influence on the mulch surface deterioration of Novomont 0.6. But for Novomont 0.7, humic acid did influence greater deterioration. Now for BASF 0.6, both humic acid and acetic acid had an effect on deterioration, but the effects were not significant from one another. For in-soil degradation, none of the products had an effect on degradation rates either. But we did learn 2 very important things from these findings. For one, you can see that in-soil degradation continued to increase over time as mulch surface area loss increased for all the BDM treatments, in particular for the Novomont mulch treatments. So this also let us know that these mulches in particular may be more suitable to use in our region. So to summarize, there was no effect of product application timing on mulch surface deterioration. Humic acid did have an effect on surface deterioration as well as acetic acid, but it was not consistent between the plastic BDMs. Lastly, the products did not influence greater in-soil degradation of any of the BDMs, but we did notice greater degradation rates in the BDMs made from starch and PBAT. I would also like to quickly mention that I used a fava bean cover crop in the field where the mesh bags were buried. And so as you can see from the photos, the roots of the fava bean crop went through the mesh bags and had an impact on BDM fragmentation. So it would be worthwhile to continue exploring in future studies. So I'd like to thank everybody in my committee and SFH team for their support, as well as the WSTA for providing funding. And I would like to thank you all for watching my lightning talk today.